Good morning, guys of grade 8. Today, inshallah, we're going to continue the first lesson of chapter 4. So we keep talking about the actual reproduction and the meiosis. So in the last session, we already just figured out the meaning of some new terms. We know what mean by sexual reproduction, then we know what mean by diploid cell and haploid cell, and so on. So, and we end that the sexual reproduction is a kind of reproduction that requires the combination of the DNA or genetic material of two parents. One is male and the other one is female. The male make a sex cell, which is sperm, while the female make another sex cell, which is the egg. Then they fertilize each other, or the fertilization process happen, in which the two cells will combine together to form the zygote. And the zygote has a full number of chromosomes due to the combination of the half genetic material from each parent. So then we said that the diploid cell, the cell that contain the full number of chromosomes, like all cells inside your body, while haploid cells, they are the cells which just have the half number of chromosomes, and the special kind of cell, which are the sex cells that just have the half number of chromosomes. That's to ensure that after fertilization, the zygote will have just the full number or the complete number of chromosomes, that to keep the number of chromosomes constant in the species. So we said that this kind or these six cells, the egg or sperm, to have the half number of chromosomes, they must be formed by a special type of division, which is meiosis. So here meiosis, that's the division which has already caused the formation of six cells. So meiosis just happen only in the reproductive cells, the cells which are already found in the reproductive organ, which means the ovary in female and testes in male. And that division lead to formation of six cells, sperm and egg, which each one contain half number of chromosome. So here, as we can see in meiosis, one diploid cell divides and make 4 haploid sex cells. Okay, so here we said that we are going to say meiosis occurs only during formation of sex cells. That means meiosis just happen in reproductive cells to produce sex cells, which means sperm and egg. So we need to clarify the difference between reproductive cell and sex cell. They are two different types of cells. Reproductive cells, it means the inner layer of cells which found inside testes in male and ovary in female. And these cells go through the meiosis to produce sperm and egg. Okay, so sperm and egg, we call them sex cells. Meiosis involves two divisions of the nucleus, while meiosis 1 and meiosis 2, which means, as we know in mitosis, we just have one division of nucleus. We call it mitosis. After mitosis, we have one cell with two nuclei. Then cytokinesis divide the cell into two new cells. Each one have one nucleus, which is exact copy like the original one. While here the meiosis, it's different, because actually we have meiosis 1, which means already the nucleus divide, and we have two nuclei, then cytokinesis that produces two cells. Then these two cells pass with another meiosis, which is meiosis 2. That means each cell will divide again. So we end up with 4 sex cells, 4 sperm or 4 egg, okay, that are produced from meiosis of one reproductive cell. So here we said that a reproductive cell go through interphase before beginning in meiosis. That means what happens. So to know this, just to find that, guys, let's see here what was going to happen. So we said, or to understand this clearly, we're gonna find here, we said, like any cell cycle, okay, we have interphase in the beginning, okay. So in interphase, the cell prepared cells for the division. So the cell will pass by G1, as we know, then S, then G2, okay. So here like this, that G1, the cell will grow and duplicate its organelle S, okay, the application of DNA. G2, the cell reach the maximum size and be ready for the division. So that's the same happen. After interphase, the cell will pass by meiosis 1, okay. So after meiosis 1, we're gonna have 2 cells, okay. Then these 2 cells will pass to meiosis 2, in which another division for nucleus. So here the nucleus will divide twice. So here just like this. So they are gonna have actually 4 cells, okay. So these 4 cells would gonna be the 6 cells, okay. So these 6 cells, if it's in male, so they will be sperm, 4 sperm, okay. If they are in female, okay, so they will be 4 egg cells, okay. So here, like that's what's happened, okay. So like this, just one cell passed by interphase, then passed by meiosis 1, meiosis 2, which lead to the end of division of 4 cells. Okay, so that's here how we got, okay, or that's how the division happens. Okay, or that's the overview of meiosis. The question is how meiosis is already done. What are the steps? How the cell go through this division? So like that, we are gonna have two types of division of nucleus, meiosis 1, or two steps of division of nucleus, meiosis 1 and meiosis 2. Actually, meiosis 2 is exactly the same like mitosis, no difference. They somehow the same, no difference between them. While meiosis 1, that's the one which have some differences than this. So each one of these, okay, meiosis 1, okay, and the meiosis 2, they already happen on steps, the regular steps which we know them. What they are, okay, so we have prophase 1, then metaphase 1, then anaphase 1, and finally telophase 1. Okay, so here that's the different steps of meiosis 1. The same steps will happen in meiosis 2. Okay, but just we're gonna give it the number 2. So here we have this happen in 4 steps. Okay, prophase 2, metaphase 2, okay, anaphase 2, and finally telophase 2. Okay, so actually the meiosis 2 exactly like mitosis, no difference. The same steps which we said in mitosis will happen in meiosis 2. While in meiosis 1 we have some differences that happen in each phase of the 4 phases of meiosis. So what are the differences? We're gonna find them, okay, together now, and we are gonna know this. So let's see how meiosis happen. So here again, we said, okay, let's see. So there are 4 phases of meiosis 1, the prophase. So the prophase, let's find what are the differences which are gonna happen here. So in prophase, the nuclear membrane breaks down, the same like mitosis as we studied before. And of course, chromosomes will have enough space to condense or to appear clearly. Nucleus also will disappear and spindle fiber will appear. So they are exactly the same like prophase of mitosis. Somehow, except one thing only, that chromosomes will not, they will arrange in pairs, or they are gonna stand in pairs. That means each 2 chromosomes will stand beside each other. Each pair, each homologous chromosomes will they will stand beside each other. So here in mitosis, actually each chromosome was just separate from the other. They don't care to stand in pairs, but here it's necessary to stand in pairs. Why they must stand in pairs? Actually, this part is not mentioned in your book, and you must add it. Okay, it's very, very important part to know it. Okay, so here we're gonna find that you will find, so in that's our cell. So we said that in prophase, okay, the nuclear membrane just that will break down, and then we're gonna find that the pairs of chromosomes will stand in form of pairs. So after prophase, of course, each chromosome is replicated. Now it comes like sister chromatids joined by centromere, so that's why it appears in X. So we're gonna find each pair, they're gonna stand like this beside each other. Okay, so that's one pair, and that's the other pair, for example. Okay, so like this, we find they are standing in pair, not separated from each other. Okay, so here it is together. We call it homologous chromosome. So like that, okay, we find these stand in form of homologous, okay, and the other changes still the same. Of course, the spindle fibers start to form, okay, that's each centriole pass to one end and start to form the spindle fiber. So the same changes like mitosis, except the only difference that chromosomes are arranged in form of homologous pairs. Why, especially in prophase, they must arrange in homologous pairs? Because actually there is a process happen, very, very important. This process, call it crossing over. This one actually is not mentioned in your book, but it's very important to know that. What is crossing over? Crossing over, it is the process in which the internal parts of the chromatids, or the internal chromatids of homologous chromosome exchange parts. How? Look with me carefully here, guys. So that's here our pair. We're gonna make it bigger just to show to you. So that the blue one, okay, and that's the red one beside it, okay. So they stand like this. Then after a while, we're gonna find they they end like that. Look with me careful here. So what's happening here, guys? They exchange part. The red chromosome take part of the blue, and the blue one take part of the red. These parts are exchanged. They switch it. Okay, they switch it. These parts. Okay, so we call this process crossing over. Okay, so this is very important process. Why? Because actually, while changing this part, it means we change some genes between the homologous pair or between the homologous chromosome. So here, this chromosome, okay, now actually is a new combination of DNA. It's not like the original. And this one also is new combination. It's not like the original. So every time during formation of sperm cells in male or egg cells in female, the sperm cell or the egg cell will have new combination of DNA, which absolutely different than another possibility, or which is different than other sperms and other egg. That's why every time your father and mother, they give birth, your brothers or sisters, you are not identical, although you have the same DNA from the two parents, but each time you have this kind of variety. Why? Because each time while your father making sperms inside his body, okay, this crossing over happened, and while your mother making eggs inside her body, so this crossing over already happened. So every time they pass a new combinations of DNA in their sex cells, which make a new variety of living organisms. So here, that's what's happened in the first step, which is the prophase. So that's prophase 1, okay? So that's the difference, okay? So after prophase 1, we get the second step, which is metaphase. As we remember, what I mean by meta, we say that metaphase it is the phase which the chromosomes are gonna line up, okay? So the same actually will happen here, but we still have the same difference, okay? That's here, the cell and chromosome, they will line up in form of pairs. That means they are gonna line up in 2 lines, not just 1 line. So here like this, we're gonna find they will line like that, okay? Let's see here, okay? And that's because the change, okay, that's happened, the crossing over. And also the same will happen to the other pair. So here like this, they line up. Why they line up? They line up because of the effect of the spindle fibers. So the spindle fibers hold them like that, okay? So they line up in 2 lines, okay? Not 1 line as usual in mitosis. No, just they line up in 2 lines. So here we see that homologous chromosomes line up in 2 lines at the center of the cell. Then after metaphase, we pass to the next step, which is anaphase. Okay, so the anaphase 1, what will happen in anaphase? The same, or as usual, as we know, what will happen? We see the spindle fiber will shrink and they pull the chromosome apart, okay, or to the 2 sides of the cell. So the same will happen here, but actually instead of separating chromosome, they will pull 1 complete chromosome. So that means the chromosome will not divide, okay, but just they're gonna. So here in this case, we're gonna find after a while, after the spindle fibers shrink like this, they become small, okay. So we're gonna find. Sorry, let's repeat this, guys, okay. So here. Still elongate a little, okay, and we find. So the spindle fiber will pull, okay, just the complete chromosome. So here we're gonna find this. The red will be here, okay, and the green of course will be here. So of course the exchange is still found, but actually it's hard to keep drawing it. So just know it here, and that's 2, 1, and here pink one will be here. Okay, so they they just pull a complete chromosome to each side. So here actually the chromosomes are not separated as usual as we studied in mitosis. No, here still the chromosome still replicated, and the one replicated chromosome is going to be pulled to each side. Then we pass to the last step, which is telophase. And in telophase 1, what will happen? Okay, so the opposite of prophase. So we are gonna have a nuclear membrane, nucleolus, and spindle fiber disappear, and we're gonna find that inside each nuclei we have the half number of chromosomes. So here that will be this one, and that's inside this chromosome, and here we're gonna find this one will contain the green and pink one. Okay, so like this, okay, we got this. So if we compare, and of course after that, cytokinesis will happen, and finally we end by 2 cells. So here like this, okay, we could find that if we compare, we start this cell, how many chromosomes? Here we have 2 pairs, okay, we have 4 chromosomes, and we end here. So here we have 4 chromosomes, okay, that means 2 pairs, okay, but we end here inside each one, just we have 1 pair or 2 chromosomes only. Okay, so like this, we decrease the number of chromosomes into half in each one cell. So each one here will have just 2 pairs only, okay, of chromosome. So like this, the end or the result of meiosis 1 that we have a cell that have the half number of chromosome. But be attention that the chromosomes is still replicated, and that's not normal. The chromosomes must separate or back again to be like a thread-like structure. So that's why we need meiosis 2, which is exactly like mitosis, no differences at all, okay? So here, like this slide represents the differences between meiosis 1 and the mitosis or meiosis 2. That's here the difference. First, in prophase, that chromosomes the chromosomes arranged in pairs. That's to allow crossing over process to happen. Then in metaphase, they line in 2 lines, okay, because they already still pairs, okay, not 1 line like mitosis. In anaphase, we separate a complete chromosome. We don't cut the chromosome. We just pull 1 complete replicated chromosome. In telophase, okay, the nuclear membrane will form 2 nuclei, but each nucleus contain the half number of chromosomes. Then cytokinesis will happen, producing 2 cells. Each cell have the half number of chromosome, which is still replicated. So that's why we need to go through meiosis to divide it or to just return it into the normal single chromosome. So back to our presentation here, okay? So that's here the prophase. Metaphase, as we can see, just here they are lined in 2 lines. Then in anaphase, the homologous chromosome separate apart. That means each one we will pull, okay, a complete chromosome. Then in telophase, as we can see here, the nucleus contains 4 chromosomes, while the 2 new nuclei, each one contains just 2 replicated chromosomes. So that is meiosis 1, the differences between meiosis 1 and while then they will pass to the meiosis 2 directly. That means the 2 cells which are produced there are gonna pass to prophase 2, which exactly here we can see the chromosomes not arranged in pairs, so no crossing over will happen or nothing, exactly like mitosis. Then just metaphase, they will line up as we can see in 1 line. Then anaphase will separate them, and finally telophase. So the end we have 4 cells, okay? Each cell contains the half number of chromosomes, okay, like the original one. These 4 cells actually they are gonna be the 6 cells. That means in male they will be sperms, in female they will be egg, okay? So here that's the difference, okay, or that's show the steps of meiosis, okay? So like this we know the difference, okay, between meiosis and mitosis. Then we ask a very important question. Why meiosis is very important? So actually meiosis is very, very important because meiosis cause, okay, or formation of the 6 cells which have the half number of chromosomes, haploid cells. Why it's important? Because after fertilization, the zygote will have a complete number of chromosomes. So meiosis help to maintain the number of chromosomes constant in a species, okay? So that's very, very important process, okay? Plus that, meiosis causes genetic variation because the crossing over process, as we explained during prophase 1, crossing over process happened. So always new combinations of DNA will form during formation of 6 cells. That's why it causes, okay, that we never have to have the same DNA. So like this here, guys, that's the meiosis and the importance of meiosis. I hope it's very clear for you. Please study it very well, and you have any question you can ask anytime. Next time, inshallah, we're gonna continue our lesson and just we are gonna okay revise this part again. Thank you, guys.